The NoRCEL session is supported by a network of researchers on chemical emergence of life. And every month we invite a speaker to talk about the raw topics in biology. And today we have a pleasure in having with us today Dr. Francisco Prosdocimi. But before I introduce Dr. Francisco Prosdocimi, Dr. Martin Dominic would like to say some words. Martin, please.
Okay, thank you, Savio. So, hello everybody, and welcome. It's great to see you all here today. I'm Martin Dominic. I am the president of the Network of Researchers on the Chemical Emergence of Life. And if you hadn't heard about NoRCEL before, NoRCEL aims to foster a global community of inspirational thinkers on the topic of the chemical emergence of life and the universality of biology, recognizing that there are lots of different aspects to it. And maybe the solution to many questions is not found within one specific discipline. So we have to bring people together with quite a different expertise, and we need to listen to each other. And we need to think. We need to think very carefully, and we need to explore new directions that probably many people who are too deep into thinking in just one direction won't be able to go. And that's where our strength is. So if you haven't heard about NoRCEL, you can check us out on our webpage, norcel.net. And you're completely welcome to join. You're most welcome to engage in further activities of NoRCEL. This is just one of them, where we have these monthly sessions with very interesting speakers. And talking about very interesting speakers, I hand you back to our friend Savio.
Thank you, Martin. And now I would like to introduce my friend, my collaborator, and my scientific brother, Dr. Francisco Prosdocimi. Francisco, thank you so much for accepting my invitation. Please share your screen and go ahead.
Okay, Savio. Okay, Martin. It's a great pleasure to be here. I hope you are listening to me well. I'll share here my screen view. Please let me know if you can see it.
Yes, perfect.
Okay, very good. So, yeah, as Martin was just talking, I'll just introduce myself briefly. I'm a biologist. I am a geneticist. And together with Savio, we have been studying the origin of life in a theoretical perspective. Sometimes we do experiments as well, and generally bioinformatics experiments. But our point of view is much more of a theoretical biologists and theorizing and thinking about the origin of life and trying to propose new models for the origin of life. So we have been publishing dozens of papers in the last decade. And we have now solid, what I consider as a solid working model that I'll present to you today. I will focus this lecture in these 4 great eras of evolution of life in Earth that we have divided in these 4 eras that we call the prebiotic age. This was the first era. Then the age of Fuca, the second era, the age of progenotes, third era, and the age of organisms, the 4th era. And I'll be talking to you the main issues, the main questions, and the main controversies that we can find among researchers working in all these stages. This level of controversies that I put here, it's like a subjective analysis that I did myself. And I consider the prebiotic age quite well resolved nowadays. I mean, there's still lots of controversies, of course, I'll be telling about, but it's probably after the age of organisms is the most understood way and eras that we can understand how life originated in Earth. Then the age of Fuca, there are lots of controversies, and the age of progenotes that I'll be talking to you here more deeply. Also, there are lots of controversies, mainly because here in the age of Fuca, as we'll see, there are different concepts of what life is, and we have to understand and to agree about what life is if we don't want to make any controversy. And the age of progenotes, I put here there are more controversies because I think nobody talks about the age of progenotes, and I think like we are the first that are actually theorizing about the age of progenotes. So there are lots of controversies because nobody are talking about it. And I think it's the most important era that we as researchers in the origin of life, we should focus. So I'll start with the prebiotic age, okay? In the prebiotic age, we have, therefore, the early Earth, okay, the Adian Earth, the Earth before biomolecules. We have what we call the prebiotic chemistry, okay? And this was inaugurated by the classical work of Stanley Miller in 1953, where Stanley Miller proposed for the first time his experiment, the Urey-Miller experiment, and proposed the origin of amino acids. He proved from very early that amino acids could be formed in this earth, this prebiotic earth. Okay, and then there are lots of questions in this prebiotic age. The main ones being the question against the panspermia against the primordial soup. Was everything inside Earth, or something came from the meteorites, from the comets to seed the life here? Okay, and also the discussions here are mainly the prebiotic biology discussion that is about exactly the origin of those biomolecules, mainly the origin of amino acids and the origin of nucleotides. Okay, of course, also the energetic issues related. So the astrobiology at the prebiotic Earth, I divided this prebiotic age in two stages. At the first stage, we have the formation of the old, what I call the old primordial soup, with or without the aid of extraterrestrial source. I don't think this is actually a question. Doesn't matter exactly whether everything was inside Earth or something came from comets. It may be, for me, it's likely that some things may have come from comets, but it's not an important issue in my point of view. Okay, so we have the formation of the first biomolecules in Earth, okay, and the formation of the first building blocks for biomolecules that are the origin of amino acids, the origin of ribose, the nucleobases, and the phosphate backbones for nucleic acids, and the origin of the first nucleotides. This is necessary. There would be no life if these molecules could not be made in the prebiotic soup. Okay, so these are necessary steps for the next development of life. And there was also in this prebiotic soup some cycling of molecules that will form the prebiotic pathways that are naturally occurring by the physical chemical issues happening in the prebiotic soup, and the cycles will generate other chemicals due to the regular period of Earth rotation and the Earth rotation in between among itself, the rotation of the Earth, and also the translation around the sun generate regularities that would bring molecules, inorganic molecules at this time to occur. And then what I call the second stage of the prebiotic age. In the second stage, we would have already an abundance of molecules and biomolecules in the prebiotic soup. And then in the first stage, the biomolecules were forming, and the second stage, they were abundant in the prebiotic soup, okay? Also, they were abundant and they were cycling, they were exchanging radicals and chemicals and parts of atoms with other molecules, and allowing the sort of complexification of chemical routes happening there in the prebiotic soup, the prebiotic compound, the prebiotic metabolites, and also we produce what we will call the protobiotic pathways. We make a differentiation between the prebiotic pathways and the protobiotic pathways that I'll be explaining to you in the further slides. Also, in the second era, there was the origin of amino acid binding, okay, and the formation of peptides naturally occurring. That this was demonstrated also by Stanley Miller, okay, he found amino acids and a couple of peptides, and also the origin of nucleic acids. This is the most controversial issue in the prebiotic age, and I'll be talking about this, the origin of RNA self-replication and the development of an RNA world. In our model, these are also necessary steps for life to merge, okay? So in this sense, we have published this paper that's named the Prebiotic Chemical Refugia, on which we propose different refugia, different places, different microenvironments that were prone for a couple molecules, the biomolecules, to originate. Actually, this paper is a review on which we reveal the most likely scenarios for the origin of phosphates, for the origin of phospholipids, for the origin of purines, pyrimidines, for the origin of amino acids, for the origin of carbohydrates like ribose, that was very important for the formation of nucleotides, okay? And it's probable that some of these molecules, they were originated in different refugia, different microenvironments that we call the prebiotic chemical refugia, on which there were sufficient amount of their building blocks of atoms and other molecules that could ease the formation of each of the parts of the nucleotide, of the amino acids, and of membranes a little further, okay? So these microenvironments we call the prebiotic chemical refugia, inspired by the theory of the Pleistocene refugia for the origin of mammals, okay? Well, for the origin of amino acids and peptides, there's a consensus that they are easy to be formed in the prebiotic chemistry with the chemical proposal by Stanley Miller. That is even nowadays the most likely chemical atmospherical scenario for early Earth. But then for the origin of nucleotides, the controversies are much higher, okay? How the nucleotides were formed in early Earth. So some of people say that comets, meteorites, and asteroids seeded the planet with these building blocks, mainly the building blocks for nucleotides, because it's an agreement that the building blocks for proteins were available in Earth, okay? But in any case, for the ones considering the entrance of those molecules by comets, meteorites, and asteroids, there is a problem. That's the controversy of anti-heating. because molecules are subject to intense heat and pressure due to friction when they came from these meteorites. And this friction, this intense heat, might have destroyed or altered the delicate organic molecules, including carbohydrates that were on these star bodies, okay? So I think it's more likely that they were already present in early Earth, okay, in the primordial soup, in the reducing environment that was rich in organic compounds. And generally, the entrance and the panspermia hypothesis is considered as a non-necessary supposition, even if it can facilitate the formation of some molecules. Also, another controversy and another proposal for the formation of nucleotides in early Earth is that the nucleotides were formed in the hydrothermal vents, that they are extreme environments. They are geological features happening in the ocean floor, in between the tectonic plates, where the tectonic plates meet. Okay, there is this volcanic activity that brings from the center of the Earth heat and minerals and molecules, okay? But also this is a very extreme environment with high temperatures, and we imagine that there were happening some chemiosynthesis, okay? They don't depend on sunlight, but they have lots of chemicals that could produce some molecules like important for making nucleotides, okay? Okay, and then there are the ideas of the shallow ponds. The shallow ponds are these small lakes that were present in the early Earth, and they were the water on these lakes evaporated, and then it rained, and then it formed a lake again, and it evaporated, and then this cycle of evaporation and watering would bring a constant flow of nutrients and some sort of concentration of the molecules that were available around there, that when the lake starts to evaporate, the molecules would get very concentrated and would bind to each other. And this is nowadays, the main theory for the formation of purines and pyrimidines, okay, for the nucleobases of nucleotides. The shallow ponds are considered nowadays as the most likely prebiotic refugee on which this nucleobase could be formed. Okay, lots of paper on it. Also, there's the idea that life, that life would be created or facilitated. Biomolecules could be formed in clay minerals because they could have acted as templates or catalysts for the formation of some organic molecules. They are protected environments, but there are also criticisms saying that they may not have enough energy, and although they can be considered another refugia on which nucleotides could start to be formed. Okay, so this was stage one, on which the first biomolecules should be formed. Then we got to stage two of the prebiotic age, on which we have already an abundance of biomolecules. With this abundance of biomolecules, the molecules started to bind to each other. We imagine a scenario with an abundance of amino acids, and this abundance of amino acids would also produce an abundance of peptides, okay, small peptides that would bind, amino acids would bind to each other due to physical chemical forces, okay, and would start also to bind another molecules in the prebiotic soup. As we know, proteins and peptides, they bind easily. They have this amino, this carbonic acid one side and the amino group on the other side, amphoteric molecules capable of binding lots of different molecules. and then the peptides created by physical chemical blind forces in the prebiotic soup would therefore start to cycle, okay, and to bind to different molecules and to cycle as well and produce different sort of molecules. And there are lots of papers saying also that most or many of the basic pathways in biology, like the glycolytic pathway and the citric acid cycle, they could happen in prebiotic earth without the presence of any biological catalyst. Just by physical chemical forces, these molecules, they would change to each other and produce a cycle, okay? This would be an Eigen-like cycle based on the idea of Manfred Eigen of this early cycles happening in the prebiotic earth, okay? Also, with this stage 2, with the abundance of biomolecules in the prebiotic age, we will have this world of sugar bases, phosphorus, and nucleotide production. Nucleotides must have been produced easily in some prebiotic refuge, in shallow ponds, in hydrothermal vents, somewhere they were produced in high amounts. This is also necessary for our model. And this high production of nucleotides would then generate an RNA world of self-replicating, self-catalytic RNAs that were capable to produce, to replicate themselves, okay, and produce new molecules. So the world of peptides and metabolite, it was based in the regularities of the astronomical cycles, of the circadian cycles, day, night, and the moon cycles, the month cycles, and the year cycles that would allow the production of biomolecules in some sort of regulated environments in the prebiotic refugia, okay? And these organized forms of interaction would then produce cycles of molecules, and the cycles also start to cycle together with peptides to produce what we call the prebiotic and the protobiotic cycles, okay? Then the RNA world is also a necessary step for us. We imagine the abundance of nucleotides and RNAs. The replication of RNAs were happening by plus minus. You have a strand of RNA, and then another nucleotides would bind here, and will be growing from 5' to 3', and by this plus minus binding, the RNAs would replicate themselves. And now inside the RNA world, there would be natural molecular selection, and natural selection would select molecules that replicate faster, that have higher stability, and also molecules that have higher copy fidelity, higher fidelity of copy, okay? And when this RNA world started to bind with amino acids, and this RNA world got together with amino acids and peptides, this brought us to the origin of life. The RNA world is not life, and also the peptide world is not life. But then when they got together, we have the emergence of the PTC, the peptidyltransferase center, and we start with the first universal common ancestor, that is FUCA. Okay, so when these two worlds of replicating RNAs and peptides start to bind together and to talk together, life emerges in our model. Okay, so what's life for us? Life is a process, okay? A process of chemical symbiosis, a process on which a chemical code, an organic chemical code is formed, okay? and this chemical code is the origin of the PTC that will evolve to the ribosome and evolve to the FUCA. Okay, this table is from our manuscript organic codes, and then in my opinion, this is our most important contribution to the scientific knowledge up to this moment. FUCA is a concept we created, the first universal common ancestor, and FUCA is born with the origin of biological codes. Okay, the origin of the genetic code is the maturation of FUCA. And then we have proposed all these concepts, the tRNA core hypothesis, the chemical symbiosis, FUCA, and the emergence of FUCA, and from FUCA, how we evolved to the ribosome. So this is a Savio's idea, the tRNA core hypothesis. We imagined an RNA world on most of RNAs were probably in a sort of tRNA form, tRNA shape, the clover leaf structure, okay, because it's the most stable form of simple and small RNAs. The tRNA has 3 hairpin loops that form this shape of a 3-leafed clover, the D arm, D loop, the anticodon loop, the variable loop, the T loop, and the acceptor stem. And the tRNA were cycling and replicating in the RNA world. And at some point, tRNAs start to bind together with 5' to 3' concatenation, and the tRNAs concatenated themselves and produced what we call the PTC, the peptidyl transferase center. And the peptidyl transferase center, a proto-PTC, is what performs, what makes the life to emerge, actually. This is the chemical symbiosis theory. Okay, so for us, life emerges when RNAs start to bind and talk by a sort of chemical communication to amino acids and peptides. So biology for us is based on the information exchange among molecules, chemical communication. And the chemical symbiosis theory is a specific branch of biosemiotics that's named cold biology, okay? For us, our model equals the relevance of nucleic acids and peptides to the origins of life. And our proposal is a proposal of a ribonucleoprotein world, RNP world, okay, and solves an ancient chicken and egg problem of the geneticists, considering that nucleic acids were more relevant to the origin of life, and biochemists that consider that peptides were more important for the origin of life. For us, they are equally important, and without the RNAs or without the peptides and proteins, life wouldn't emerge. So they are very important, and life is based on a ribonucleoprotein process, okay? So our chemical symbiosis reinforce the relevance of symbiotic forces for the biology since its first breath, and was a symbiosis among nucleic acids and amino acids and peptides that make life to emerge. Okay, so we thank Lee Margulies. We published this paper in 2020. A Margulian view for the emergence of biological system. That is our theory of chemical symbiosis. So the chemical symbiosis have produced what we call a first proto-organism, the first entity, the first biological system that was truly biological. This is FUCA. FUCA is the first universal common ancestor, okay? And it's the great-grandmother or the great-grandfather of LUCA. It will emerge much earlier than LUCA and was the emergence of the first gene, the first molecular communication between nucleic acids and proteins. And FUCA was born as a proto-PTC. as an earlier version of the peptidyl transferase center, that is the catalytic center of the ribosome. Okay, FUCA for us, we have a paper describing better. This was a concatemer of proto-tRNAs. The proto-tRNAs concatenated, and Savio demonstrated this in a paper on which he aligned he calculates the most likely sequence of proto tRNAs for each amino acid, and he concatenated the proto tRNAs, and he realized that the concatenation of transfer RNAs ancient that were produced by a model to make ancient molecules, when they are concatenated, they are more than 50% identical to the PTC of a bacteria, in that case, the Thermus thermophilus. Therefore, we realize that the FUCA is made by concatenated tRNAs, and we know which tRNAs are these. This is the proto-tRNA of proline, the proto-tRNA of tyrosine, the proto-tRNA of phenylalanine, the proto-tRNA of glutamine, and the proto-tRNA of glycine. When these proto-tRNAs were concatenated, we were able to form a PTC that has a 3D structure very similar to the PTC observed nowadays in ribosomes, and they are also two different structure, one for binding amino acids and another to facilitate replication. And this was published in Life 2020. Also, thanks Miriam also that helped in this work. We found the sequence of all bacteria, all the PTC of all bacteria, the PTC of all archaea, and we find that also they were very similar. So after the PTC, the ribosome was formed by a lot of different phases on which the ribosome, the higher subunit, the largest subunit of ribosome was assembled, and then the small subunit was assembled, and then we don't know what happened for the genetic code to be finally assembled, but we know that this was the most important issue in the age of FUCA, the self-organization and the self-assembly of a stable genetic code, a stable code on which triplets of nucleotides can bring specific amino acids. Okay, this defines biology for us. This is the most important event this is a maturation of FUCA. FUCA is born as a PTC that binds randomly amino acids, and it maturates as a sophisticated form of protoribosome capable to bind amino acids specific to triplets and codons in the RNAs. So we don't know how this code was generated. We have some hypotheses, but we know that the person that will discover how exactly the genetic code were made will probably earn a nice award on biology, because it is a very important issue, and we know, and we are searching for it, but it's very complicated problem. Okay. So FUCA therefore starts with the emergence of proto-PTC, and it finished with establishment of a working genetic code. Okay, and we have doubts how perfect was this code at that time, but it was perfect enough so that the genes could arise, and so that organized peptides could be formed. And then we go to the following phase, that would be the third era, that would be the era of progenotes. Okay, therefore we start the prebiotic age. With the Fuca, we have the age of Fuca, the second age. Then we go to the progenote age, and the progenotes will therefore generate the organismal age. We have our models is in constant improvement, okay? And as we are publishing new articles, we are improving the model, okay? So I have already talked to you about the prebiotic age and the age of Fuca. Now we go to the age of progenotes. What happens between the formation of the ribosome and the formation of cells, or the formation of organisms? What happens? The genomes must have been assembled. The protein folding was complexified. Protein binding evolution, the assembly of biochemical pathways, and the origin of protoluca and the luca. Okay, so the progenote era, that was my title in this lecture, is a paper we are writing it now. It's very advanced, and I have been writing it on doing this lecture as well. So the progenote era starts with a genetic code working quite well, a translation system working quite well, and a world of self-reproducing ribonucleoprotein particles, okay? So it's an RNA world that was symbiotically linked to peptides and metabolites in the prebiotic soup. The progenotes, what were the progenotes? The progenotes is a concept given by Calvus in a paper of 1998. And we have also, he doesn't tell exactly what was a progenote. It was a precellular entity that has lots of changing of DNA, like lateral transference of genes. And we theorized it better. For us, it's a sort of naked self-reproduction ribonucleoprotein particles, okay? We know that these ribonuclear particles, they can form sort of a cytoskeleton, the ribonuclear proteins. Inside cells, we have in the cytoplasm some sort of compartmentalization of the cell given by ribonucleoproteins that they work as a cytoskeleton, okay? So we imagine this happening without cells, in a colloid, in a water environment, okay, on which the ribonucleoproteins, they provide some aggregation, okay? And this is also studied in the field of people that study prion-like proteins, that they cause aggregation, and ribonucleoprotein particles also turn aggregation, okay? So in this environment, this prebiotic refugia on which ribonucleoproteins are in the median and provide some sort of cytoskeleton and some sort of specific microenvironments, ribonucleoprotein particles were evolving, were replicating, were binding to molecules in the primordial soup, okay? And they were evolving, and they were changing, exchanging material, genetic material by lateral transference. And the main issues in this era is the origin and the maturation of genomes, how the genomes got bigger, okay? How we got from Fuca to Luca. This is the progenote. The progenote starts with the Fuca, and the progenote era finishes with the formation of Luca. This is the progenote era, okay? Protein binding evolution and the evolution of biochemical pathways. So the genomes start to maturate, okay? We have different progenote populations evolving independently, and these progenote populations, they start to concatenate with each other to form large RNA genomes, okay? And in the age of progenotes, this is a picture of our next manuscript, on which we'll show how this progenote and these genomes are being assembled at that time. And then the progenotes start to get these bigger genomes, and the metabolism is born also at that stage. At that stage, the genes are producing proteins, okay, in progenotes, in naked. The progenotes use ribosomes available in the media, and they translate their information, okay? We have the complexification of protein binding and catalysis, the development of new peptides and new 3D structures for proteins, and different progenote populations evolve different sorts of molecular bindings. Some populations evolve carbohydrate binding peptides. Some populations evolve amino acid binding peptides. And some populations evolve nucleotide binding peptides. This would generate in some populations the development of carbohydrate pathways, such as glycolysis. In other populations, the development of biosynthesis and degradation of amino acid. And some populations, the development of biosynthesis and degradation of nucleotides. Okay, so this was happening also in the the prebiotic without any catalyst before, like the idea that there were cycling of metabolites in the prebiotic soup, and at some point some peptides will bind to these metabolites in the prebiotic soup, and they will facilitate the transformation of A to B with a time that's smaller than the time necessary for physical chemical forces to modify A to B. With the age of binding of peptides, this time is diminished, and this is exactly the origin of catalysis, of biological catalysis. Okay, also peptides binding to B will diminish the time to transfer B to C. This is the origin of catalysis. We are always criticized in this point when the people say, oh, but there were probably lots of metabolites and lots of cycles that happen in prebiotic earth that they don't happen nowadays in organisms. Of course, there were lots of chemical cycling in the prebiotic earth that were not incorporated in the biological organisms. And this is exactly the difference, how we differentiate what we call the prebiotic roots against the protobiotic roots. Okay, so the prebiotic roots are the pathways that were not incorporated in the biological organisms, while the protobiotic pathways, they were incorporated in the biological organisms. Okay, so this is described in this paper of us, Entering the Labyrinth. Okay, we published it last year in Biosystems. All right, so we at this point we have stepwise formation of Luca's genome. Luca's genome was then formed. The genome of the last universal common ancestor was done with the concatenation of the most well-succeeded progenotes genomes. Okay, probably needed that progenote population were deeply mixed and evolved in different environments, different organisms, different pathways were probably evolving in different microenvironments, in different prebiotic refugees, okay? And then at some point, this got together and the genomes were concatenated and generated this highly well-succeeded organism, proto-organism that was still a progenote, that was Luca, the last universal common ancestor, okay? At that point, we can also imagine the origin of biological encapsulation, because viruses, for us, some virus, of course, not this virus that we found today in Earth, but probably lots of viruses, they evolved before cells, okay? And they evolve by proteins that fit in together, okay? So the origin of biological encapsulation allowed progenotes to travel along unhospitable and microenvironments and colonize other microenvironments. And these different progenotes that were living here and there, with the encapsulation, they could travel and got together and then cross these energy thresholds, and then produce the LUCA genome. Okay, viruses were probably originated by simple proteins that fit in together and allow the formation of simple capsids capable to hold this genome of a progenote and then allow them to travel. And we said in this article from 2019 that viruses do not need cells to replicate. Viruses, they need ribosomes to replicate, not cells. Viruses are progenotes that were encapsulated without ribosomes, and later delivered this naked, these genomes that were inside them to naked ribosomes for expressing and replicating their content. An evidence of the virus early model is that capsids are order of magnitude simpler than proteolytic membranes, and the viruses probably originated multiple times in evolution, being an evolutionary stable strategy and not a sort of dependent entity from cells. Okay, viruses, they have encoded genes and they speak the language of biology. Therefore, viruses are alive. Okay, so we have the prebiotic era, then the era of Fuca, then the progenotes, and the inside the progenotes and viruses live it together, and then just later the cells came into the code. We have published this paper last week about exactly the origin of viruses. Okay, we said in this article that the viruses are the most abundant ecological system in Earth and cannot be considered as mere infectious agents. Some viral clades evolved certainly from progenotes and predate cells. Okay, the origin of viruses allowed progenotes to become encapsulated and conquer new environments. Viruses interact with cells to be able to access their ribosomes nowadays, and most viruses are symbiotic organisms important to the homeostasis of environments and holobionts. Therefore, the concatenated out of most well-succeeded progenotes genomes generate the genome of LUCA, of the last universal common ancestor. There was not a cell, there was a progenote. LUCA was the first prokaryote. Most of the pathways, biological pathways we know today were in LUCA, but not DNA biosynthesis and not membrane biosynthesis. LUCA has no DNA probably and has no membrane also in our model. Okay, so the sons of LUCA are the cells. From the LUCA that was a progenote prokaryote, cells arose. Okay, so we call the Castor and Pollux. That is the next lecture I'll give here in the NoRCEL in September. I'll tell you about the sons of LUCA, and the sons of LUCA are the first bacteria and the first archaeobacteria, okay, on which the DNA biosynthesis and the lipid biosynthesis evolved independently. The DNA biosynthesis in bacteria use a set of enzymes that are not homologous to the set of enzymes used by archaeobacteria, and also the biosynthesis of lipids. Okay, we know nowadays that the membranes are 40% of the biological membranes are proteic. This means for us, this is an evidence that membranes evolved by lipid binding proteins. Okay, membrane is lipoproteic, and the membrane evolved from proteins, not from lipids. okay? So once the LUCA genome is formed, we got to the age of organisms when the membranes will be formed, okay? We start then the age of organisms. From LUCA to the cellular organisms, we got the abundance of cellular life that we know and we see today in Earth, okay? The origin of membranes happened 2 times. The membranes from archaea are very different, okay? They have a ester linkage, they bind a different carbon in the glycerol, and they have a side chain, while the membrane for bacteria and eukarya are also completely different. So they have different pathways on which the membrane evolved independently in bacteria and archaea, evolving from different populations of the LUCA, of a prokaryotic LUCA. Also, the DNA biosynthesis in bacteria and archaea, they use different sort of enzymes. They are not homologous, and they were probably originated independently as well in bacteria and archaea, okay? And of course, eukarya, they are a modified, a sophisticated form of an archaea, at least in the genome. So to finish this lecture, I'll tell you the history of life in Earth. Okay, we published this paper, Origin of Life, drawing the big picture, inspired in the last talk I gave here in NoRCEL. Okay, I think it was Martin that said, oh, we need a big picture. And then I said, okay, I'll write a paper named The Big Picture. And this is the big picture. We have the prebiotic refugees in the prebiotic age, okay? We have abundance of amino acids, nucleotides, an RNA world working. And then we have the origin of life when FUCA is first proposed by the binding, the PTC, an RNA capable to bind amino acids together and create peptides. Then we have the maturation of the protein synthesis apparatus, okay? The evolution of progenotes, okay? The evolution, the origin of protein capsids, the origin of LUCA, and then the age of organisms, the origin of cells, 2 times, one for archaea and one for bacteria. And this is our model. This is another picture of a paper we published last year, the RNP world. The ultimate essence of life is a ribonucleoprotein process, and where we say also the age of molecules, the age of FUCA, the age of progenotes. Here, the viruses for us are in the same, originated in the age of progenotes, okay, and then the age of cells. Here is the model. We have the prebiotic chemistry, then the chemical symbiosis, the emergence of the genetic code, the evolution of progenote, the initial assembly of genomes and biochemical pathways, the capsid world with the simple capsid being created, the evolution of capsids, creating new forms of capsids, the origin of LUCA, the origin of membranes, and the cell-based world. This is a stepwise, gradualistic model that we produced based on the literature and our thoughts. So this paper will be coming in the next months, the age of progenotes, the origin of genomes, and the assembly of biochemical pathways. And this is the main issues I will be describing. We will be describing me and Savio in this paper that is about to be submitted. So thank you so much for listening. It was a great pleasure to be here. Thanks, Savio, for the long collaboration. He was the one that invited me to work in the origin of life when I was a simple genomic guy and bioinformatician. I'm very glad for you, Savio, for inviting me. It has been a great satisfaction for me in my career to work with you and to develop these models. That is very inspiring for me. I thank Sohanjita and Martin Dominic and all the NoRCEL staff for the invitation. Thank also Marco Jose, Miriam and all his group, all the co-authors of all papers I described here. I thank the copyright owners of the pictures I used here, and the Midjourney bot on which I created pictures for this lecture as well. And I thank for the financiers, FAPERJ, CNPq, and the Federal University of Rio de Janeiro